Okay, so I'm actually so excited for this video. I have worked on this for actual days. Oh my gosh. So I am an artist and I like to draw things, and I'm going to be going over their evolutionary history using both these drawings but also some physical examples of them. So here I have a couple of brachiopods that I want to show you, and what you'll notice is that they look very similar to clams. And I also have I'll get one more example here just so you can see like diversity in size and shape and whatnot. But I do have a clam here. This is a clam known as Keone. It was unfortunately killed by a gastropod. So they look a lot like clams, but these guys are very distantly related. Clams are part of the phylum Mollusca, while brachiopods are their own unique phylum, Brachiopoda. And this convergence on a bivalved body plan to filter feed involved twice. Well, I think maybe around 3 times really, but that's besides the point. It evolved a lot of times. But they are their own unique group of organisms, and I just like them a lot because they're very, very pretty and very, very diverse. So I will be referring to this chart throughout the video, and I just want to show you how to read it so that we're not confused later on. Okay, so this here is the geologic timescale. So we have the Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian, Triassic, Jurassic, Cretaceous, and then I just kind of bunched the Cenozoic together in one big group. Up here I have the big 5 mass extinctions. This is the end Ordovician, the end Devonian, the end Permian, the end Triassic, and the end Cretaceous. I put a star here because this is the worst biodiversity crisis in the Phanerozoic. This entire eon is known as the Phanerozoic. This is the worst biodiversity crisis that we've had in the last 500 or so million years. And it is integral to the story of the brachiopods, which is why I put a star next to it. Something else that you'll notice is that the vast majority of these species, or I shouldn't say species, of these orders are completely extinct. They're completely extinct. So we have 5 living orders: the lingulids, the graniids, the rhynchonellids, the pterobrachioids, and the thesideiids. I think I'm saying that right. Thesideida. What is called the Thesideida. Alright, the Thesideida. Awesome. There's 5. But there's around 19 to contentiously 20 orders here. I should say contentiously more. There's a lot of orders that I didn't list because I didn't really think that they were like that relevant to talk about, so I didn't list them. But around 20 orders of brachiopods, so 15 of which are completely extinct. So the vast majority of their diversity is completely locked away in the fossil record, which is why I find them so interesting. So there's around 385 to 388 living species of brachiopod, and they aren't particularly diverse. They're typically relegated to deep water or polar environments. There's one genus called Lingula, which is quite common in the Indo-Pacific, but typically in brackish water and estuarine environments. But it's an exception to the rule. So, in terms of fossil species, conservatively, there's around 12,000 fossil species. But I've seen that number go all the way up to 30,000. So they are incredibly diverse in the fossil record. So the question is, what happened? And I will lead you through this story as we go through. And I guess we'll just start off with their evolutionary origins. So brachiopods are a part of this informal grouping known as the Lophophoridates. They get that name because of a lophophore, which is a feathery filter feeding apparatus that you see here, and there. The brachiopod sister clade is known as the Phoronids. They are very worm-like in appearance and still exhibit this lophophore. Like if you were to rip open a living brachiopod, you would see a lophophore as well. So the theory is what we call the brachiopod fold hypothesis, right? So the Timodiids are kind of in this outgroup of the brachiopods. They aren't true brachiopods, but they're likely very closely related to the first brachiopods. Wolfangella is likely related to the ancestor of the very first brachiopods, and it's this worm-like creature covered in these sclerites, these scales made out of chitinophosphate. So chitinophosphate is what we call a weakly biomineralized shell. So chitin is a very durable biomolecule, which is the same stuff that insects and arthropods form their skeletons out of, and phosphate in the form of apatite, which is the same mineral that your bones are made out of. So it's very weakly biomineralized, but it still counts as biomineralization, right? So you have this worm-like critter, like Wolfingella, that is. Hold on. I am filming this in the middle of the day, so it's just is what it is. Anyways, Wolfingella, this ancestor, right, probably resembled this, and eventually attached itself to a hard substrate like its eccentrica, right? And as time goes on, these plates, these scales, become larger in size but reduced in numbers. So then you get something like Tenwalina, and eventually something like Macrina, where there's only 2 large plates. But the entire organism is hosted completely inside. And this is why so if you were to look at the internal anatomy of a brachiopod, it's very, very different than that of a bivalve. I will say that even I thought that brachiopods were clams when I first found out about them, and so did the first scientists and paleontologists. But again, I want you to understand that it is a completely different evolutionary origin than that of the mollusks. They are their own unique group of organisms. Alright, so there's also some other terms that I want you to understand before we get into this. So the first is the dorsal or the brachial valve. It gets its name because of the lophophore, the arms, I guess, of the brachiopod. And the ventral or the pedicle valve, it gets its name because of this pedicle here. The pedicle is a fleshy stalk that attaches a brachiopod to a substrate like mud, hard ground, or even other shells. But so again, I need you to understand at least those 3 terms. The dorsal or the brachial valve is the top of the brachiopod. The ventral or the pedicle valve is the bottom of the brachiopod. The pedicle is where it attaches itself to a substrate. Another thing that divides brachiopods and bivalves is their musculature. Bivalves only have adductor muscles, which is what closes the shell together. So that's why when you do a clam bake, the clams all open up because the muscles relax. But brachiopods actually have 2 sets, the adductors and the diductors. The diductors are what open the shell, so they have muscles to close the shell and muscles to open the shell. Now this does vary. The more basal brachiopods, like the linguliforms, actually have oblique muscles. They don't have diductors, but it's basically, for all intents and purposes, just understand that their musculature is also quite different. Other things I want to talk about. The beak is, this is a really simple one, or this one. The beak is this kind of outgrowth right there on the edge of the shell. The commissure is this imaginary line between the 2 valves. Oh, that's another thing that you can use to define brachiopods and bivalves. So a bivalve actually lies on its side. This bivalve is officially on its side. This is right side up for a bivalve. But this is the top and the bottom, or I guess, yeah, I believe so. I believe that's the top and the bottom of a brachiopod. Right, their line of symmetry runs through the shell, but not along the commissure because they're uneven on each side. But a bivalve symmetry runs through the commissure, but not along the shell like a brachiopod. Right, I'm hoping that makes sense and that you can see what I'm talking about. Right, bivalves are not symmetrical through the shell. Anyways, the fold and the sulcus. So the fold is this large, what's the word I'm looking for? I guess rise here on the top of the shell. The sulcus is this invagination here on the underside of the shell, and this is very indicative of ringonellate brachiopods. The last thing I want you to understand is shell microstructure here at the bottom. So this has to do, so ringonelliform brachiopods form their skeletons out of calcite, which is a variety of calcium carbonate. Excuse me, sorry. So there are three kinds of shell microstructures depending on whether or not the calcite, there's like I guess pores in the calcite, right? So punctate would mean that there are pores and holes in the calcite structure. Pseudopunctate just means that pseudopunctate means that the lamellar calcite sort of pushes through but not all the way, while impunctate just means that there's no holes in the microstructure of the shell. Alright, and this is going to be important because this is how we define some of the different groups of brachiopods. For example, pseudopunctate you will only see in strophomenate brachiopods. Alright, so I guess we can finally start talking about their diversity. I first want to talk about lingula. I feel like lingula is a really good example of your typical brachiopod. So the interesting thing about lingula is that it's a living fossil. We have species that look very similar to lingula that date all the way back to the Cambrian period. It is a very old morphotype, and you can see all of these basic features, like the bivalve shell and this pedicle that the organism uses to root itself down into the substrate. So let's get started. So the first major group that I want to talk about, I'm going to get rid of these actually, I don't want to use them. So we're going to start with the early Paleozoic. So in the early Paleozoic, this is going to be from the Cambrian through the Ordovician period. We're not going to be able to get them all in there, so we'll just move it down as we go through. We start off with the linguliforms, right? So the linguliforms, all the way over here, have two major classes: the lingulodons and the pteranodons. The pteranodons are a really cool group of the linguliforms. So again, the linguliforms all form their shell out of chitinophosphate, and they don't really have a great fossil record. They're either preserved as carbonized impressions or as here, actually I have one right here. This is orbiculoidea. This kind of like gossamer brown kind of looking shell. Now this is a really bad example of what a linguliform looks like, but generally if you see that in the fossil record, you know that you're looking at a linguliform. But the pteranodons are kind of this ecological blip, or I guess kind of a short story in the major story of the brachiopods. They're not particularly diverse, but they're really interesting because, well, first of all, they have these really, really pretty, intricate shells, but they also have a lot of features that are very reminiscent of the Ringonelliform brachiopods, which is, the important group of the brachiopods. They have these hinge teeth. So hinge teeth are actually what we use to define some of these different groups. So I'm going to use Keone as an example because you can see it really, really well. But lots of bivalve organisms have what we call a tooth and socket, where you have an outgrowth on the hinge and a socket on the other side, and this kind of locks the shell together. I'm not going to put that back, actually. This kind of locks the shell together, and I do have an example of that for a brachiopod. This is some kind of orthid, I believe. But if you can see it, those tiny outgrowths are the hinge teeth, and this is what locks the shell together. But we only see this in the Ringonelliforms and the pteranodons. But we think that this is a different evolutionary origin than the ones that formed the Ringonellids. So there's this really archaic grouping known as the inarticulates and the articulates, and that's where that comes from those hinge teeth. But we call that a paraphyletic grouping, where it doesn't really represent any natural relationships. So we got rid of it and made this new grouping here. But the pteranodons end up going extinct at the end of the Ordovician, and that's kind of the end of their story. The lingulodons, though, are very important in the Cambrian period. So actually, I am going to talk about this because this is very relevant to the entire story of the brachiopods. But there are that was number two. There's three evolutionary faunas proposed by Jack Zipkowski. So there's the Cambrian fauna, the Paleozoic fauna, and the modern fauna. So what this refers to are groups of organisms that are ecologically dominant and diverse during a particular period in time. So the Cambrian fauna, these are your linguliform Paleozoic fauna include the rhyngonelliform brachiopods. All these guys here, they're ecologically dominant from the Ordovician through the Permian period. And the modern fauna includes things like crustaceans, snails, and bivalves, and marine reptiles and all this sort of stuff, right? That are dominant from the Triassic onwards. The other Paleozoic fauna I should probably mention are things like rugose and tabulate corals. I actually have a tabulate coral right here. I think this is Alabora. This is a Paleozoic fauna. There are, some other ones actually that I'll bring out. We have some rugose corals here. These are also Paleozoic fauna. Here I have Pentramides. This is a really cool echinoderm. It's a stalked echinoderm, so I don't have the rest of the stalk. It disarticulates pretty easily after death, but this is a stalked echinoderm known as Pentramides. It's a blastoid. There's other, so crinoids are also another example of the Paleozoic fauna. One other, actually, just while I have this out, I actually want to show you this too. I have a Eurypterid. This is also, I guess, a Paleozoic fauna in the sense that they're ecologically dominant, but we only really see these guys in the Silurian period. They're only really dominant in the Silurian, but I just think this fossil is really, really cool. This is of Eurypterus ramipes. I think this is from Baltica. I can't remember exactly. Anyways, back to the brachiopods, the interesting stuff, right? So the lingulids and the acrodretids, I have two examples here. Here's Lingulella and Acrodreta proper. These guys are the most dominant brachiopods in the Cambrian period. They make up something like 70% of the brachiopod diversity. But they kind of taper off into the Ordovician with the evolution of the rhyngonelliforms. Again, all these guys here. Again, they're lingulids. They all kind of have this sort of shape to them. There's another group called the Decidids, which have these like silicic vesicles in them. Let me see if I can find it. Here we go. But I wouldn't say that they're like particularly interesting. The Decidids in here. And they grow these vesicles on top of their shell made out of silica. I know they're kind of weird. But anyways, yeah, I think that they're still pretty identifiable to each other. Acrodretids are generally going to be these cup-shaped things made out of chitinophosphate. So if you see one, you'll know it's an acrodretid for sure. There's no doubt about it. But there's also another group, this is Phonotratina. Sorry, I'm like going through this so quickly because there's a lot to go through. Where is it? Where is it? Oh, it's on this page. Here we go. They're also kind of an evolutionary blip. They are very spiny. They have these kind of like pitted structures to them, but they end up disappearing in the late Silurian period, and they're just, I don't know, I think that they're really cool, but we have a lot to go through, and if you want to do more research on them, go do more research on them. Anyways, the next group I want to talk about are the craniiforms. So these guys they don't have a pedicle, actually. This is Craniops. This is a really good example of a typical craniate brachiopod. They encrust over other shells and hard ground substrates, but they do not have the articulate condition like the rhyngonelliforms. But one group that I think is very, very interesting are the trimerellids, this group here, because they are the only arachnidic brachiopods. So why is that important? Here I have two examples. Here's Trimerella and Eodonopolis, and I also have this sort of cross-section of Trimerella here. So you can see inside of the shell. But anyways, why is that important? Well, the calcium carbonate has two major polymorphs in our ocean, aragonite and calcite. And really the major difference is just how the crystalline structure is expressed, right? But the majority of brachiopods biomineralize using calcite. So it's really, really weird that these guys use aragonite. And they were very, very diverse in the tropical belt in the Ordovician period, dominantly in Kazakhstan. And so they're a really good index fossil for not only the late Ordovician, but also for a tropical distribution of rocks. But they are almost annihilated in the end Ordovician extinction, and they kind of straggle onwards to the Silurian before finally going extinct. But Cambrian period. Here's Styrophora, an early orchid. So do the pentamerids, these guys here. And I'll show you all of these as I go through all of this. But these guys are the Paleozoic fauna. They experience the majority of their diversity from the Ordovician through the Permian. They're really, really cool. And I think what I'm just going to do is just sort of talk about them as we go through time. So we'll start in the Ordovician period. So in the Ordovician period, we see the dominance of a group known as the orthids. So here I have an orchid here. There's another one there. This is actually a Devonian orchid, but I'll still talk about them anyways. But so orchids are a little strange in the sense of there isn't really a whole lot of great defining features for them. They are possibly paraphyletic as well, because every other brachiopod order, we believe, may have evolved from the orchids. But it's really hard to say, because I know I made this distinction here, but it's brachiopod taxonomy is always in flux, because again, the majority of their diversity is locked away in rocks. The only thing that we really have to go off of is morphometrics, and it's not really a great system anyways. So for right now, this is how I'm dividing them up. Anyways, orchids are generally going to be very biconvex, meaning that you have really just two large. It just kind of looks like this. It's sort of, I guess, globular in shape. They have a very prominent beak, like that, and what we call costate, which is this really fine ribbing. However, there are exceptions to this. Here is a really common Ordovician orchid known as Platystrophia. It has a very distinct fold in sulcus. Of course, I only have the bottom half of this thing, but this is the pedicle valve. A very prominent fold in sulcus. It's plicated, which is really just very intense ribbing. But yeah, the orchids experience their peak diversity during the Ordovician period. The other group that experiences peak diversity is going to be the Strophomenids. Now, I only have Devonian Strophomenids, but they generally kind of look like this. Yeah, they're these like really D-shaped shells. We kind of call them the scallops of the brachiopods. They were free-living. They did not have a pedicle in their adult stage, and they just sort of clapped their shells together and swam around like on SpongeBob, right? So let's see here. But there is a distinction I want to talk to you specifically about, and this is between the Strophomenids and the Orthotetids. Right, so here we have, I'm going to use these two as examples. These are not these species, but I feel like it's really easy to see the difference, right? So pretend that this guy is Strophomena and this guy is Shelwionella. Strophomena is a Strophomenid and Shelwionella is an Orthotetid. Well, they look very similar to one another. They have a very strophic hinge line, which means a straight line. They're very D-shaped shells, so what's the difference between the two? And this has to go into shell microstructure. So Strophomenids were generally, I believe, just pseudopunctate. You would only find they're only pseudopunctate, sorry, I apologize. While Orthotetids were impunctate, pseudopunctate, and punctate. And this is really the major difference of why we made this distinction between the two. And again, I don't understand it entirely. Again, a lot of this stuff is just research that I have looked up that is available online. I don't actually study these guys for my job, so I just think that they're fascinating. I would love to work with them one day, though. Oh yeah, Orthotetids also have an extended cardinal process, which is this thing up here. Like, it's just sort of very wing-like in shape. But yeah, it's I guess jargon, I suppose, in terms of the difference between the two, because they just look very, very similar to one another. But Strophomenids have their peak diversity during the Ordovician period. They experience a huge drop in diversity. The Ordovician mass extinction and just sort of struggle onwards until the end of the Permian. Let's see here. Other major groups that appear during this time are the pentamerids, the rhynchonellids like Hiscopecus, and really all of these major orders, the spire bearers. And I'm just going to have you take this in because it's easier to talk about them when I talk about just know that the Rinconelliforms appear in the Ordovician and rapidly diversify into all of these major orders. Right? Okay. There we go. So, the mid-Paleozoic is the time of the Rinconellates. So, we have a couple of different orders here that I want to touch on. The first here is Dicelesia. It's an orthid brachiopod, but it's also what we call bilobed. And this actually appears 3 times total in the brachiopods, once in the Devonian Dicelesiids, a second time in the Permianellids, which I'll talk about later, and a third time in the Cretaceous Bicopids, which is a pterobrachylid group. Anyways, this is the time of the Rinconellates, and it's going to be really easy to kind of just go through this and talk about that evolutionary history as we go through. So the orthids are still around in the Devonian period, but they're not particularly as dominant as they were in the Ordovician. They experienced a huge diversity drop. But into the Silurian period, we see the dominance of this group known as the Pentamerids. So Pentamerids first appear in the Cambrian period, but again, they were never really taxonomically diverse, but they could be very locally abundant. So in the early Silurian period, directly after the late Ordovician mass extinction, we see the sudden appearance of these huge Pentamerid beds, particularly a Virgiana. These guys were co-supportive. They did not have a pedicle in their adult stage. They just sort of laid on top of one another, but they formed these huge beds that it's really, so if you see a lot of Pentamerids in these rocks, you're probably looking at Silurian rocks. These Pentamerid communities, though, were very short-lived. They sort of straggle on throughout the Silurian, and Pentamerids do not make it past the Devonian extinction boundary. I should probably explain. Pentamerids get their name because of their internal structure. There's a specific thing I'm looking for. Here it is. There is this thing called a septum and a spondylium that kind of divides it into 5 chambers on the inside of the shell. 1, 2, 3, 4, 5, not equal chambers, but 5 chambers, and that's where they get their name from. They are impunctate shells that are generally very biconvex, and again, when I say an evolutionary blip, it's an unfortunate way to refer to them, but the orthids and the pentamerids give rise to essentially every other order. So we think that the orthids evolved into the pentamerids, and from the pentamerids we get the Rinconellids, which then evolved into everything else, right? So the Rinconellids are these really nut-shaped shells, like this guy here. They have very strong, what we call plication. Here's uncinulus, and they're kind of nut-shaped. They look kind of like just a nut. They also have a prominent beak, and they were never really, I guess, dominant in the sense, in the same way that all of these other brachiopods were, but they were very, very consistent. So since their evolution in the Ordovician period, they've been around, and they're one of the few surviving orders of brachiopods that we still have. But they're, I should say, easily identifiable, because there are some Rinconellids that kind of break this body plan. But generally, if you see strong plication, like really strong ribbing, kind of a short hinge line, what we call astrophic hinge line, it's kind of sort of like this, like the pentamerid, they have an astrophic hinge line. You're probably looking at a Rinconellid. But from the Rinconellids, we get a group known as the spire bearers. These spire bearers are the most important group in the Devonian period. So they first appear in the Ordovician, but start to rapidly diversify into the Silurian. So the first group to appear are the Atripids. So here I have a couple of Atripids. Well, they're probably all the same species, to be honest with you. But I have a few Atripids. Let me see if I can find some more. I'll want that one later. Let's see here. Here's another, I believe. Yeah, we'll just use those two as an example, right? So they are the first spire bearers. So what this means, and what this is referring to, is this is basically a calcified slinky inside of the shell. These spire bearers used to be united under one order, these Spiriferids, but for understood in my opinion, or at least not very easy to understand. So there are a variety of flow dynamics depending on the shape of the shell and the direction and shape of this spiral brachidium. The reason for this is that the lophophore would wrap itself around this chalcidic spire, and again, in filter feed using that, right? But there's two different flow dynamics. There's the active and the passive flow dynamic. So a active flow dynamic would mean that the cilia on the lophophore would beat in unison, and water would come in through the side and out through the front, right? So the brachiopod would filter food out and oxygen from the side and push out all of its waste products out to the front. However, under a passive feeding regime, it actually goes the opposite way, right? Where the water goes in through the front and out through the sides. So how does that work? Well, the brachiopods that lived in very high energy environments probably were more passive filter feeders. So they took advantage of all that wave action and storm energy, and the water would naturally move itself through their shell. They didn't have to do any work except just open their shell. But again, it's very contentious because, again, there are no living spire bearers except for the pterobrachiopods, but the pterobrachiopods actually have a different kind of spire. It's called a loop brachydia, and so that's why it's kind of difficult to determine exactly the flow dynamics of how this worked, because we don't have any living analogs. So even though the pterobrachiopods evolved from the atripids sometime around the Middle Silurian period, they have their own unique brachidial structure. But here's Rensselaria. It's a very common brachiopod, or very indicative brachiopod of Devonian rocks. But this is the time of the spire bearers, so we think that the spire actually probably evolved 3 separate times. We think that because the Reganellids had what we call a cruce on their shell, and that this cruce is from which the spires all derived from. The other major group is going to be the spiripherids proper. So they have these very wide wing-like shells. This is actually a Permian version. Yeah, they're really, really pretty shells, in my opinion. But how do I even describe these guys? Yeah, they're really good index fossils for the Devonian period. They're common throughout the late Paleozoic, but the Devonian period is peak diversity of these spire-bearing brachiopods, particularly of these spiripherids. The other major group is the athirids. So this here is actually, well, I think this is a late Paleozoic variety. Let's see if there's one that's a little better preserved. I think we'll have to make do. So, I definitely have one that's better preserved than that. I know that for sure. Oh, come on. I have so many brachiopods in here. None of them are the ones that I'm looking for. Here's a Thyrus proper, I believe. There's one that has a really obvious pedicle foramen. We'll use this one. So the athirids are this really kind of globose, rounded, very what we call unplicated. So they don't have this ribbing on their shell. They also have a very obvious pedicle foramen. So you see that hole right there at the end of the beak. That's where the brachiopod would attach itself to a substrate. But again, these were also spire bearers. These spire bearers, so we have our athirids, our spiripherids, and our atripids, were all very, very dominant during the Devonian period. But the end Devonian mass extinction almost completely annihilated them. So the atripids go completely extinct at the Devonian. Well, they actually go extinct before the end of the Devonian. There were actually a series of extinction events. I refer to them as the end Devonian extinctions because there's actually major pulses. So there's the Frasnian-Famennian boundary and the end Famennian, right? So the end Frasnian was the first major pulse of extinction. This is where we lose the atripids and a lot of other major groups like stromatoporoid sponges or stromatoporoids, or there's a different way to say that. There's based on where you put the accent. 80% of the brachiopod diversity at this boundary, which is really unfortunate because, again, this is where we experience the most diversity, and from this point is just sort of the beginning of the end for them, going into the late Paleozoic. So the late Paleozoic is the time of the Strophominates, the group that I've kind of been neglecting to talk about so far. So the first major Strophominates appeared in the Cambrian period in the form of the Billingsellids. There's a specific paper that I'm looking for because it has Billingsella on it. Here it is. I'm really only just gonna. Oh, come on. Okay, sorry about that. There was a plane. Anyways, you probably heard it. Strophic line. This is probably the first major appearance of the Strophominates in the fossil record, and that's all I'm really going to talk about them. Anyways, I just wanted you to know what I was talking about when I said that. But the other groups, specifically the productids, are what I'm going to talk about for the late Paleozoic. This is actually my favorite group of brachiopods, and unfortunately, they're also the most diverse in this particular time period. They make up anywhere between 45 and 70% of the brachiopod diversity. It's an insane amount. They're really, really diverse. So there's a couple of different groups. The first group is the Canidids. So here I have them as a lopus. I actually don't know if this is Canides proper, but this is very closely related to it. These guys are very reminiscent of the typical Strophomenate body plan. Wide strophic hinge line, very flat shells. I also want to notice that I want you to note that this is the full organism. As flat as this is both valves stuck together. They are very, very flat, very, very small organisms, but they were free living. They sort of swam along the seabed like so. But the Canidids are, I guess, the basis for which all of this other diversity. Sorry, someone was calling me. All of this other. I hope that wasn't happening long. Oh no. I hope it didn't cut out. We'll see. That all of this other diversity comes through, right? So, I guess we'll just talk about the productids next. So the productids have these really, like, kind of deep shells. They have a very flat, planar, or what we call, I guess it's convex, brachial valve, right? So it's planar biconvex, or planar convex, sorry. Anyways, or concavo convex, and all these other different things that you don't really need to worry about. Anyways, they're very deep shells, but they also have spines. The spines are what define the productids. They're very, very spiny, and they use them in different ways. So some, like the Phenocanides here, probably use them to just sort of elevate themselves above the substrate so they wouldn't sink on soft mud. But they then diversified into all of these different forms that we see here. So Lena productus specifically used its spines to actually encrust over hard substrates like bryozoans or corals or hard ground, and I just think that's so cool. Other functions for these spines, like Poloni productus, it was probably used for defense against predators. The interesting thing about these spines is that they're actually hollow on the inside, so they could continually grow out these spines as they grew. So as they, I guess I want to say as they grew with them, right? Wagenoconcha is really interesting because it used its spines kind of like roots, right? It was what we say called a semi-infaunal. So it was sort of half buried in the substrate and used its spines to sort of root itself down into this soft mud. Others, like Pouchus spinifera, we think that some of them you probably used it like stilts. So they didn't want to be in the soft mud. What they would do is just sort of elevate themselves above the sediment-water interface, so that their lophivores don't get clogged by all that fine-grained mud. So lots of different varieties in form and structure. I also don't have a whole lot of species of these because productids are very hard to come by, especially these guys, the Lytaniidine. They always have the most difficult names. Litanidina. Yeah, anyways. So I mentioned this earlier when I talked about bi-lobed structure. Probably had something to do with the efficiency of filter feeding. There was one lophophore per lobe, and it could probably very, very efficiently push water in and through the shell. Another group, the Lytenids proper, are really, really weird. So they have this really frondose brachial valve, and this we don't really know what it was for, really. Some have proposed that it was comb-like support for the lophophore. Others think that maybe it provided a substrate for endophotosymbionts. So like lots of different marine organisms like corals and clams have photosynthetic relationships with dinoflagellates. And we think that maybe they probably might have as well, but unfortunately that leaves no geochemical trace on the skeleton, so we can't test for that directly. The other weird part is they had really weak musculature. If they did have muscles, they weren't very well developed. So some of these actually, they didn't even close their shells at all. So exactly what they were doing, we're not entirely certain. What we do know is that they were very, very common, particularly in the Tethys and in the Texas reefs. The Permian Texas reefs are one of my favorite places in the whole world. But they were very, very common. We just don't really know what they did, you know? So these Leptodus, this one here, they were encrusters. They kind of grew over one another. Others, like old Hamina here, were free living on the soft sediment. But yeah, these guys are really weird. The last one I really want to talk about is the Ricthothenids and the Orthotetids. And oh my gosh, so these guys are just phenomenal in their own sense, because these are the coralline brachiopods is what I like to call them. So they're just really cool. Let's see here. I'm hoping I'm not forgetting anything. Yeah, anyways. They are very, very what we call heteromorphic, right? They don't look like any of these brachiopods that I've shown you thus far. They are very, very different. So what was going on? We also don't really know. So again, there's no living representatives or really anything that looks like these today. So it's really hard to get an exact picture of how these guys lived. So the first Ricthothenids probably looked like Lenoproductus, like Allistegis here, using their spines to grow on top of and cement to hard substrates. But then you get this really interesting growth pattern where the brachiopod is then completely enclosed within its own shell. So here is the brachial valve, the top valve, and here is the pedicle valve on the bottom. The entire brachiopod is completely inside of this cup. So the question is, like a shape like this is used to maximize the flow dynamics of water, right? So if you're completely cutting yourself out of the water, how did you filter feed? Well, some think that they used the brachial valve to kind of pump water in and out, and this would have allowed them to differentiate between different particle sizes, right? So smaller particle sizes probably wouldn't be pulled down, but larger particles would be swept up in this current and allowed them to feed on it. So Cocinerina specifically was a free-living brachiopod. It lived in soft mud substrates, while Herclocestria, Cyclocantharia, and Cestropoma all were encrusters. I actually have one here. It is really, really tiny, and I have to be really, really careful. It's a silicified fossil of, I think this is Cyclocantharia ginglorum. But you can see all those tiny little cups that the brachiopod is living in. Anyways, I have to be really careful putting that back. Anyways, yeah, these guys used their spines sort of like roots. They were able to root themselves down onto each other and into substrates. It's actually really cool. It's so, I mean, how do you even get a growth pattern like this from something like that? It's so interesting. It's so interesting to me. Yeah, so similar flow dynamics, but the Cyclocanthariidae are kind of the weird. So they had spines at the top of their cups, which we think may have added for extra filtration. But in an extreme example, we have Cestropoma. It is completely encased inside of the shell. So there's kind of this calcite mesh over the top of the cup. I kind of drew it here so you can kind of see it. The brachial valve is completely inside of the cup. It is completely enclosed inside of itself. Why did it do this? We don't really know. Again, maybe something to do with predation. Maybe it was to help filter out larger, smaller particles coming in. We don't really know. Again, these guys are really enigmatic in terms of their exact ecology. Another one I wanted to talk about was Gigantoprodactus. So this is, I will say, speculative. There aren't really a whole lot of great fossils to use as a reference for drawing Gigantoprodactus, but there are related species that I used. So this is Gigantoprodactus. This is the largest brachiopod that ever lived. It came in at a whopping 1 foot long. So yeah, not particularly large, but Gigantoprodactus lived during the Carboniferous period, right here, somewhere around here, right around the Mississippian Pennsylvania boundary, like between them, right here in the middle. Anyways, it was huge. Why? Well, the Carboniferous was a time of rampant oxygen concentration. So today it's around 21%, but during the Carboniferous it was all the way up to 35%. So for organisms that have a passive breathing system like terrestrial arthropods and these marine invertebrates like brachiopods, they were able to get huge. So this is why you see those, those giant dragonflies, giant millipedes, but you also see gigantic brachiopods during the Carboniferous, like Gigantoprodactus. We also think that maybe, like giant clams, they possibly could have been photosynthetic, but again, we don't really know. Like there's this correlation between large body size and marine invertebrates and them being photosynthetic, like corals and clams, right? They're able to achieve that large body size because they have endophotosynbionts. And it's possible that Gigantoprodactus did as well, but again, there is no geochemical trace left on the skeleton, so we can't really say that for sure. So maybe it was only due to the rampant oxygen concentrations, or maybe because it was able to photosynthesize. As well. I don't know. So much cool stuff going on. So the last couple I wanted to talk about was Cyndalia. It was a part of a group known as the Gemelloroidae. The Gemelloroids are really, really weird. So they were initially placed within the Rhynchophenids due to their weird shape. Cyndalia, specifically Rudistiformis, gets its name because it resembles a group of Cretaceous clams known as the Rudists. But again, convergently evolved. They have their own unique musculature that places them within the brachiopods, and it was a mud. They were co-supportive. They kind of just laid on top of one another. They weren't really rooted into anything, but we thought they were Rhynchophenids. But analysis of the shell microstructure actually places them as derived orthotetids over here. Specifically within the superfamily Derbyoidea. Here is Derbia itself, which I feel like if you were to look at this and say, I think that this came from this, I feel like that's not too much of a stretch in my personal opinion, but I don't know. I'm just an amateur, right? Anyways, the Permian period is so even though the Devonian was peak brachiopod diversity, this is where they have their most taxonomic diversity in terms of the number of species. In the Permian period, this is where we see the most ecological variability within the brachiopods, specifically within the Strophomenates. So the question is, with all of this diversity that I've been talking about, right, what happened to them? Why are they no longer ecologically dominant? Well, that has to do with the end Permian mass extinction. So I'm going to pull up one. I'm just going to lay these down so we can go back through it all. The end Permian mass extinction was devastating for the brachiopods and a lot of other Paleozoic organisms. So we see the complete extinction of. Oh, sorry. Let me pull it out. The trilobites go completely extinct at this boundary. We lose all of our rugose corals. We lose all of our blastoids. And we lose around 98% of our brachiopods at this boundary. They were almost completely annihilated at this boundary. Again, this was the worst biodiversity crisis in the last 500 million years. There was a lot of volcanic activity going on in Siberia at the time, huge lava flows, and widespread ocean anoxia and acidification, and lots of organisms were just not equipped to deal with it. I mean, we lost almost all, even iconic species like snails. We almost lost all of our snails at this boundary. We lose almost all of our echinoderms at this boundary, 98%. So in total, averaging out the number of species lost, 96% of marine life is completely obliterated at the end of Permian. this is why we see such stark contrast between organisms that were dominant in the Paleozoic and those that were dominant in the modern fauna, because almost all of them were annihilated in this extinction event. It's really unfortunate, personally, but yeah. So in contrast, though, the bivalves only lost around 69% of their diversity. There was a study put out by, I forget somebody, but there was a study put out where they looked at the rediversification rates during the Triassic period between bivalves and brachiopods, and what they found was that they were actually rediversifying at equal rates. But the thing is that bivalves simply had more diversity to work with, and so overtook the brachiopods in the Triassic, finally ousting them from their dominance on the benthic sea floor. So that's why they got pushed out into these deep sea environments. Another group I want to talk about, the Thesidaida. They're really weird. They're like minuscule, really tiny. They're encrusters, and they suddenly appear in the Triassic, maybe from the spider bearers, but I don't really want to talk about them because, again, they're not really ecologically important. But what I do want to say is that we lose a lot of the traditional brachiopods, the basal ones here, the Ordovician boundary. We lose not really too much at the Devonian boundary. We lose some of those more archaic brachiopods like the pentamerids and the atripids, and then we lose almost all of them at the Permian boundary. So again, all the classic Paleozoic ones we lose at the Permian boundary. So strophometids, orthotetids, orthids almost go completely extinct. There is some Lazarus taxa of the productids in the Triassic period into the very earliest Triassic, but they don't last that long, and they finally succumb to extinction. However, the spider bearers, the sporiferids, and the ephirids make it all the way to the Jurassic period. In fact, they actually survive the End-Triassic mass extinction event, which is a similar extinction to the End-Permian. Widespread volcanic activity camp. The Central Atlantic magmatic province releases a lot of CO2 and greenhouse gases into the atmosphere. Widespread ocean anoxia. And they survive it, and then they go extinct in the early Jurassic. Why? Well, there was actually another period of widespread ocean anoxia known as the Toarcian anoxic event, in which the splitting of Gondwana, so from South Africa and Antarctica, caused another ocean anoxic episode, and by this time the sporiferids and the ephirids were kind of pushed into those deeper water polar settings, and so they did not fare that well, and they finally go extinct. So yeah, the story of the brachiopods, in my opinion, is quite tragic. They had a lot of diversity. They were really, but they just, the odds were stacked against them. Unfortunately. And yeah, they're almost completely extinct. Or they're unfortunately probably on their way out. But I hope what I was saying made sense. I know it's a lot of information, a lot of jargon, but I really do find them super interesting, and there's still parts of me that's like, I didn't really explain it that well, or there are things that I got wrong, but I feel like this is a really good general guide to the diversity of brachiopods, and I really hope you enjoyed this video.